Hi, my name is Sarah Weiss, and I'll be presenting on Nisar Shajini. So Nisar Shajini was an assistant professor in the Department of Obstetrics and Gynecology at the University of Florida when he committed his scientific misconduct. He had a Bachelor's of Science in Cell Biology from the National University of Tehran in Iran, and then he got his PhD in Cell Molecular Biology from the University of Southampton. So most of his research was on uterine leiomyomas and related endometrial issues. So a leiomyoma is a benign tumor of a smooth muscle cell, and this can be anywhere in the body as long as there's smooth muscles. For example, it can be in the small bowel or the esophagus, but it's most commonly found in the uterus. So this develops specifically in the myometrium, which is the thick middle layer of the uterus. Leiomyomas are caused by high levels of female steroid hormones such as estrogen and progesterone. These high levels of hormones stimulate cell division and increase certain growth factors. Most of these do not turn into cancer, but it can lead to risk of infertility, miscarriage, and other pregnancy issues. Risk factors are going to be the reproductive age, which is because of those high levels of steroid hormones during this time. Also race, it's been found to be higher in African American women. So Dr. Shajini's research examined the expression of proteins and microRNAs in leiomyomas and endometrial cells. So he specifically wanted to look at how the formation of tumors is associated with the expression of microRNAs. He also wanted to determine what role connective tissue growth factor has on transforming growth factor action. So TGF-beta is overexpressed in fibrotic tissue like uterine leiomyomas. It is a key regulator of cell growth and apoptosis. And the main reason that he wanted to look at this is because CCN is believed to be a downstream regulator of TGF-beta. So to do this, we're going to look at myometrial and leiomyoma smooth muscle cells, and we had them isolated from premenopausal women, and then these cells were cultured and transfected with pre-MIR21 and anti-MIR21. And then also the expression of CCNs and TGF-beta1 and TGF-beta2 were measured in leiomyoma and myometrium smooth muscle cells. And to measure these expression levels, real-time PCRs, Western blotting, and immunohistochemistry were used. So Dr. Shajini did do data fabrication and data falsification. So for data fabrication, the bar graphs that he used in many figures in many of his papers were made up data points and made up standard errors. Unfortunately, this was done in 8 of the 9 papers that he published. And then he also falsified experimental groups by changing their identifiers. So this gave a false significance to the effects of different treatments on protein expression levels. And this was actually done in 6 and 9 papers that he published. And then also he changed the order of time sequence. So there was an obvious trend in the bar graphs where the values varied in a very predictable way. However, when they looked at the actual written data that was collected, the same trend was not seen. And this was seen in 2 of the papers that he published, 2 of the 9. And then so as we look here, he was actually guilty of falsification and fabrication in all 9 papers, and all 9 of them were retracted. And also this was in 3 grant applications. So in the first figure on the left-hand side, this is relating to paper 1, but the data and error bars were completely made up, so he just made these up and didn't actually perform these experiments. And then in the figure next to this, relating to paper 2, we can see that the data was actually falsified by either omitting or adding certain points to get the results that he wanted. And then when we look here, he was required to retract all 9 published papers. He also was not allowed to participate in any U.S. Public Health Service research for 5 years from the date of their agreement. So after this time, though, he would be able to apply for PHS support. However, he'd be supervised and watched extremely closely. And then he also was not allowed to serve in any PHS advisory committee, board, or peer review committee for 5 years from this agreement. And then so he was retired and he no longer works at the university. However, he did give a really bad representation of the University of Florida when he did this. And then also he used about $4 million in federal grant funding. So he pretty much just wastes this money that could have been used by other researchers that were actually performing their research in a proper way and not cheating the results. And then also additional money and time was wasted so that researchers could kind of like check and make sure he was doing things right. And then also each of his 9 retracted papers had been cited multiple times. So for example, one of his 2007 papers was cited 15 times, and then another one was 74 times. So many of these were cited.